Good day, Fluorimetry Module 5. Today we are going to learn about principle involved in fluorimetry, instrumentation and applications. What do you mean by fluorimetry? Fluorimetry is an analytical method for detecting and measuring fluorescence compounds. The fluorescence compounds uses ultraviolet light stimulating the compounds causing them to emit visible light. Principle involved in fluorimetry. Fluorescence is the phenomenon of emission of radiation when there is a transition from singlet excited state to singlet ground state. You can see in the block diagram, this is the excited state and this is the ground state. And here, the molecule undergoes transition from singlet excited state to singlet ground state. The wavelength of absorbed radiation is called as excitation wavelength, whereas the wavelength of emitted radiation is called as emission wavelength. These two wavelengths are specific or characteristic for a given substance under ideal conditions. Let us learn about electronic states or concepts in fluorimetry. Singlet ground state, a state in which all the electrons in a molecule are paired. Singlet excited state, a state in which electrons are unpaired but of opposite spin. Doublet state, a state in which unpaired electron is present. Triplet state, a state in which unpaired electrons of same spin are present. Here, absorption of UV or visible radiation causes transition from singlet ground state to singlet excited state. At this state, it is not stable. Again, it will emit the excess energy and return back to ground state. To achieve this transition, there are three possibilities occur. Collisional deactivation, in which the entire energy is lost due to collisional deactivation and no radiation is emitted. Fluorescence, a part of energy is lost due to vibrational transitions and the remaining energy is emitted as UV visible radiation of longer wavelength than the incident light. Why this occurs? This is because the energy of emitted radiation is lesser than that of incident or absorbed radiation because a part of energy is lost due to vibrational or collisional process. Hence, the emitted radiation has longer wavelength than the absorbed radiation. Phosphorescence. At favorable conditions, at low temperature and absence of oxygen, there is a transition from excited singlet state to triplet state, which is called as intercrossing system. The emission of radiation when electrons undergo transition from triplet state to singlet ground state is called phosphorescence. You can see in the block diagram. This is the intercrossing system, fluorescence, phosphorescence. In the fluorescence, the molecule undergoes transition from singlet excited state to singlet ground state. Whereas in the intercrossing system, the transition occurs from singlet excited state to singlet triplet state, which is called intercrossing system. And here the phosphorescence, the transition from triplet state to singlet ground state. Next, coming to the internal conversion. Internal conversion is the radiationless transition between energy states of same spin state. Here example we can take intercrossing system. It is a radiationless transition between different spin states. External conversion, the process in which the molecule passes from a higher electronic state to a lower electronic state because of collisions with each other. It is known as external conversion. Let us study about the instrumentation in fluorimetry. Instrumentation contains the components light source, filters and monochromators, sample cells, detectors. Light source used in fluorimetry are xenon lamp, mercury vapor lamp, tungsten lamp. Filters and monochromators. In fluorimetry, primary filter, secondary filter, excitation monochromator and emission monochromator are used. Detectors. Detectors are photovoltaic cell, photoemissive cells, photomultiplied tubes. Let us see the different components in fluorimetry one by one. First one, light source. The source should provide radiation with sufficient intensity over an entire region. In such a way, the light source should be selected. Here there are three types of sources in fluorimetry. One is xenon lamp, mercury vapor lamp, tungsten lamp. Xenon lamp. This is widely used in spectrophlorimeters. In xenon lamp, the light is produced by passing electricity through ionized xenon gas under high pressure. It provides a bright light with intense radiation over a wide range. Hence, most compounds can be analyzed. Mercury vapor lamp. Here, mercury vapor is subjected to the high pressure of atmosphere. It gives intense radiation over 350 nanometers. At low pressures, this vapor also provides one more radiation at 254 nanometers. But this is preferred in inexpensive filter type fluorimeters. Tungsten lamp. This lamp has radiation with low intensity. It is used when excitation radiation is in the visible region. You can see the diagrams here. This is the xenon arc lamp. Next one, mercury vapor lamp, tungsten lamp. Next one, filters and monochromators. The function of filters and monochromators is conversion of polychromatic radiation to monochromatic radiation. In fluorimetry, primary filter and secondary filter are used in inexpensive instruments, whereas excitation and emission monochromators are used in spectrophlorimeters. Let us see the primary filter. Primary filter or excitation filter or incident light filter. The function of primary filter is to isolate the wavelength that will cause the compound of fluorescence. So it absorbs visible radiation and transmits UV radiation. Secondary filter isolates the desired emitted light. So it absorbs UV radiation and transmits visible radiation. Excitation monochromator. It selects the desired wavelength of excitation light which is focused at the sample position. Emission monochromator isolates only the radiation emitted by the fluorescent molecule. These are the filters used in fluorimetry. Next, coming to the sample cells. Sample cells are cuvettes. The function of sample cell or cuvettes is to hold sample solution. The fluorimeter cuvettes are small cylindrical or tube-like structures. They are made up of colored corrected fused glass. All the sides of cell have polished surfaces because emission measurements are made at 90 degrees angle. The path length of the sample holder is 10 mm. You can observe the glass sample cells. Detectors. The function of detector is to convert the light energy into electrical signal, which can be read or recorded. The principle involved in detector is when a radiation is passed through a sample, part of it being absorbed by the sample and rest is being transmitted. This transmitted radiation falls on detector and the intensity of absorbed radiation can be determined or displayed. And the requirements of ideal spectrometric detectors are: the detector should give a quantitative response. It should have high sensitivity and low noise. It should have shorter response time. The detector should provide a signal or response quantitative to wide spectrum of radiation received. Usually the most commonly used detectors are barrier layer cell or photovoltaic cell, phototubes or photoemissive cells, and photomultiplier tubes. First one, barrier layer cell or photovoltaic cell. Here you can observe the diagram of photovoltaic cell. This detector is used in inexpensive instrument like filter type colorimeters, fluorimeters and nephroturbidimeters. So here in the fluorimeters we can use this type of detector. What is the principle involved in photovoltaic cell? The principle here is generation of potential difference when the transmitted UV radiation falls on the semiconductor material. Let us see the construction of this detector. The detector has a thin metallic layer coated with gold or silver. You can see this is the thin metallic layer coated with gold or silver here and acts as electrode. Also it has a metal base plate which acts as another electrode. These two layers are separated by semiconducting material, that is selenium. Selenium has extremely low conductivity and electrons are immobile. That means there is no movement in the electrons. But when the light radiation falls on the selenium layer, these electrons become mobile and are taken up by the transparent metal layer. This creates a potential difference between the two electrodes and causes current to flow when the external resistance in the circuit is small. This flow of current causes deflection of galvanometer needle which depends on wavelength and intensity of radiation. So this is the working of barrier layer cell or photovoltaic cell. But the disadvantage with this detector is amplification of the signal is not possible. Phototubes are photoemissive cells. Here the principle involved in phototubes is emission of electrons from the cathode and subsequent capturing of electrons by the anode resulting in the flow of current. You can observe the construction of the detector. The detector is composed of an evacuated glass tube. This is the glass tube. And here it consists of a photocathode, consists of wire anode or collector anode we can call. The photocathode is composed of elements of high atomic volume like cesium, potassium or silver oxide which can liberate electrons. And also composite coatings like cesium oxide, silver oxide can also be used. It increases the sensitivity of the detector. And here coming to the working. When light radiation falls on cathode, it causes flow of electrons towards anode and produces a current proportional to the intensity of light radiation falling on the detector. The signal from the detector can be amplified using the circuit. Phototubes have better sensitivity when compared to photovoltaic cells and hence more widely used. Photomultiplier tubes. This detector is most sensitive of all the detectors, expensive and used in sophisticated instruments. The principle involved in photomultiplier detector is multiplication of photoelectrons by secondary emission of electrons. And here in the construction, the phototube is present in a vacuum tube. It is fixed which receives the transmitted radiation from the sample. The multiplication of electrons is achieved by using the photocathode. This is the photocathode you can observe. And a series of anodes called diodes. These are the series of anodes. The photomultiplied tube can use up to 10 diodes. Hence, even 200 times weaker signals can be detected. The diodes are maintained at 75 to 100 volts higher than the preceding diode. Coming to the working of the photomultiplier tube. When the radiation from sample falls on the cathode, then it will emit electrons onto the diode. Some electrons from the surface from the diode gets emitted. These electrons get bombarded on the next diode surface due to higher potential. The transfer of kinetic energy from the bombarding electrons results in the emission of even more electrons from the surface of second diode. The same procedure keeps on happening and electrons keep on multiplying from diode to diode. Then finally, all the electrons hit the anode or electron collector. Therefore, the signal is displayed in the readout device. So, these are the detectors used in fluorimetry. Coming to the instruments in fluorimetry. The instruments we can observe in the fluorimetry are single beam fluorimeter. This is the filter fluorimeter, and double beam filter fluorimeter, spectrofluorimeter. You can see here the single beam filter fluorimeter, and this is an inexpensive instrument. Here the source is tungsten lamp. Filters are primary and secondary filters, and here in the fluorimetry the measurement of radiation is at 90 degrees angle. Why? Because the sample emits a wavelength which travels to the detector. The detector is usually set at 90 degrees angle to the light source to avoid any interference from the transmitted excitation light. And here the advantage of this filter fluorimeter are simple in construction, inexpensive and easy to operate. But the disadvantage is both sample and reference cannot analyze at a time, and scanning of the sample is also not possible. This is the photofluorimeter, single beam filter fluorimeter. Next, double beam filter fluorimeter. It is also similar to single beam except that the two incident beams from a single light source passes through the primary filters separately and fall on either sample or reference solution. So it is called as double beam. Two beams are passing, one for sample and one for reference. So it is called as double beam filter fluorimeter. Here also the measurement is made at 90 degrees angle. The advantage of double beam filter fluorimeter is simple in construction, inexpensive, easy to operate, and here both sample and reference can analyze at a single time. But the disadvantage is rapid scanning is not possible. Double beam spectrofluorimeter. This is the advanced instrument, and here the source is xenon lamp, and monochromators are used for the conversion of polychromatic radiation to monochromatic radiation. The monochromators used are excitation and emission monochromator, and the detector is very sensitive detector, that is photomultiplier tube. And the incident beam is split into sample and reference beam by using a beam splitter. You can observe in the diagram. And here the advantage of this spectrofluorimeter or double beam spectrofluorimeter is here the scanning of the sample is possible and very high sensitive and more accurate. These are the instruments used in fluorimeter. Next, coming to the applications involved in fluorimeter. Fluorescence analysis can quantitatively and qualitatively analyze substances based on the characteristics and intensity of fluorescence produced by the photoluminescence of the substance. So here we can study one by one. First one, qualitative analysis. Qualitative analysis means identification of the compound. So how can we identify the compound? Here, the fluorescent compounds with different structures have characteristic excitation and emission spectra. Therefore, the shapes and peak positions of the excitation spectra and emission spectra of the fluorescent substances can be compared with the standard spectra. Therefore, we can identify the compound. Quantitative analysis. Here, the concentration or amount of the sample can be measured. At low concentrations, fluorescence intensity of the solution is directly proportional to the concentration of the fluorescent substance. F is equal to Kc. Among them, F is the fluorescence intensity, C is the concentration of the fluorescent substance, K is proportionality constant. Therefore, this is the basis for quantitative analysis of fluorescence. Fluorescence techniques are also used for authentication of different food products. Therefore, we can detect the quality changes in food products. In such a way, for example, we can determine the level of lipid oxidation in foods such as fish and meat. The fluorimetry is also used for analysis of metal pollutants in aquatic environment by fluorescence methods. Fluorescence spectroscopy is also used in the determination of content of certain components in biological samples, analysis of biotechnology and immunotechnology, such as determination of DNA, antibodies, antigens. For this, various fluorescent probes are used for the analysis and detection. The fluorimetry is also used in the field of drug analysis. Fluorescence spectroscopy analysis can be used to identify the active components of drugs and also can study the pharmacokinetics, clinical pharmacological efficacy analysis. Non-fluorescent compounds can also be converted to fluorescent compounds and can be analyzed in the spectrophlorimeter. So, all type of drugs can be analyzed here. For example, you can see diphenylhydantoin. It is a non-fluorescent compound, but it can be converted into a fluorescent compound upon oxidation with potassium permanganate in alkaline medium to benzophenone. Here the benzophenone is the fluorescent compound. Methyl dopa. It is a non-fluorescent compound, but it can be converted into fluorescent compound by oxidative and alkali rearrangement, which will give a dihydroxyindole derivative, which is a fluorescent compound. Thiamine. It is a non-fluorescent compound, but it can be converted into fluorescent compound upon oxidation with potassium ferricyanide in alkaline medium and extraction in isobutanol. It will give the fluorescent compound that is thiochrome, which emits the blue fluorescence. Next, fluorescence indicators. Here, the intensity and color of many fluorescent substances depends on pH range and hence are used in acid-base titrations. You can see the fluorescent substances, their pH range and color changes. Fluorescent substances are like eosin, fluorescein, quinine sulfate, acridine, 2-naphtha-quinone. And their color changes you can observe here. So these are the applications involved in the fluorimetry. So by this study, we can gain knowledge here about the principle involved in fluorimetry. What are the components of the instrument? From this, you can gain knowledge about the principle involved in fluorimetry. What are the components of the instrumental fluorimetry?